horizontal gene transfer. Now, we've talked about DNA, we've talked about replication of DNA, and when we're replicating DNA, the point is to pass it on to our progeny, right? Just like grandma's cookbook, and you want to copy it so that you can pass it down to the next generation. So replication involves copying it and passing it down. That is called vertical gene transfer from mother to daughter, from one cell to all of its progeny. That's vertical gene transfer. Prokaryotic bacteria are capable of doing a horizontal gene transfer. They actually have a couple of different ways that they can pass DNA to their neighbor. Not their progeny, but their neighbor. And this you don't see in eukaryotic cells, but you do see it in prokaryotic cells. So let's talk about this phenomenon, horizontal gene transfer. Okay, so the donor cell contributes part of its genome. And again, when we're talking about horizontal gene transfer, unlike DNA replication where we make a complete copy, this is just sharing a portion of their nucleic acids, not the entire genome. So it is different than vertical gene transfer when you would pass everything down. So there's three types: transformation, transduction, and bacterial conjugation. And this conjugation might sound familiar because we've kind of already covered this in chapter 3. So let's skip ahead to conjugation and do that one first. Conjugation, do you remember that pilus? We talked about a pili, where it can form this structure between neighboring cells. We talked about that. It's a specialized form of fimbriae, and it involves genetic transfer. And we said what gets transferred is not the chromosome, but this plasmid. So if you go back and review that, you had an F positive cell. It's F positive because it contains a fertility factor, and then that fertility factor will replicate itself and then cross over using that pilus, and now this cell will become F positive. So we talked about that, F positive and F negative. The pilus will only form with an F positive cell, will only form a pilus with an F negative cell. We talked about it getting replicated and then crossing it over. Didn't really mention that before, but there actually is some movement in this. When that pilus is formed, it actually brings it closer. For that, this process, this crossing over of the plasmid into the new cell is called conjugation. That is one way we get horizontal gene transfer in prokaryotic cells. All right, so let's go back into the other two. There is another process called transformation. Transformation is when you get horizontal gene transfer, but instead of a plasmid, the cell picks up DNA from the environment. So maybe you had a bacterial cell that died. It got chopped up, either a drug killed it or, phagocyte, whatever reason. You have pieces of nucleic acid that are in the environment. You have a neighboring bacteria cell that is able to take up that piece. Now, most cells don't do this. Most cells, the cell membrane, cell walls, keep stuff out. We don't want random pieces of DNA and getting into our cell in general. It's a lot more chance that things will get messed up than it's not. But there are a few bacteria cells that are, or species, that are naturally capable of picking up pieces of DNA from the environment. So it's just an adaptation that they have, a survival strategy. By picking up pieces of this in the environment, then they can pick and choose which ones they want to keep. Cells that can do this and take up DNA from their environment are referred to as competent cells. So again, not all cells can do this, but there are certain species that are naturally competent, which means they have the ability to pick up DNA from the environment. Now, unlike a plasmid, this little piece right here gets incorporated into the chromosome, and then it stays with it, and then it can be passed on to their progeny by vertical gene transfer. Okay, so this happens naturally for a few species. However, as scientists figured out how this worked, it triggered a train of thought going, what if I could make a cell pick up a piece of nucleic acid? And it wouldn't necessarily be a random piece from the environment, but maybe this is a particular gene that I'm studying. And it's found in people and it causes some sort of disease, or I think it does, but it's hard to study things in people. What if I take this gene and put it into maybe a yeast cell or a bacteria cell, and then I can study it better? Or maybe this is a gene that produces insulin. And we know that patients who have diabetes, if they take insulin, they can live full and healthy lives. If they take this insulin, well, it's very hard to make enough insulin to give patients who are diabetics. What if I can make like a bacteria cell produce insulin? If I can take this gene for insulin and get it to integrate in here, and then this particular cell will pump out a whole bunch of insulin protein. That's a good thing. So there are reasons where you can do that. And this is called recombinant DNA technology, where we can use this process and get a cell to take it up. Now, once that cell has taken up the DNA, we refer to that cell as being transformed. So that's where the name comes from, transformation. Once it has taken it up, if a cell can take it up, it's called competent. Once it has taken it up, it is now referred to as a transformed cell. Okay, so that's another type of horizontal gene transfer. Happens in nature, but we've taken that theory and made it work for us. What you need to know about transduction is it involves a virus. And we're going to look at this in a little more detail in the viral chapter. But for right now, this is a case of horizontal gene transfer where parts of bacterial DNA get transferred from one bacteria to another via a bacteriophage, which is a name for a virus that infects bacteria. Bacteriophage, phage is just another name for virus. Bacteriophage is a virus that infects bacteria. And you can see you've got a bacteria cell here. This bacteria cell is getting invaded by a virus. The virus is going to kill off the nucleic acid and chop it all up, kill off the cell. It's going to make a whole bunch of more viruses, and then it's going to lyse and release these virions, these viruses, out into the environment. How transduction occurs is you get a little bit of bacteria DNA that gets packaged, and it brings it to the next cell. And instead of integrating the next cell with viral DNA, it infects the next cell with bacterial DNA, and then that gets incorporated into the chromosome, and this is where you can get horizontal gene transfer.